My name is Siri Strand, and I will present our work on the Breast PreCancer Atlas from the Human Tumor Atlas Network, where we're focusing on ductal carcinoma in situ, or DCIS. To give a brief background for the Atlas, screening by mammography since the late 1980s has reduced breast cancer-specific mortality, but it's also led to more than 500% increase in the detection of DCIS. At the same time, we observe a much less increase in the detection of invasive breast cancer, which indicates that many clinically irrelevant DCIS are being detected. DCIS is treated by lumpectomy with or without radiation therapy, hormonal therapy, or even a full mastectomy, and it's widely recognized that a lot of women are being overtreated. There are watchful waiting clinical trials in progress, but there's also a strong need to risk stratify patients to identify which ones should get aggressive and which should get less aggressive treatment. There are a lot of large-scale breast cancer studies out there, but not a lot of large-scale DCIS studies. And the H-Town Breast PreCancer Atlas is presenting the largest comprehensively profiled DCIS cohort study to date, and the data will be freely available for the research community. For the Atlas, we studied two independent retrospective cohorts of patients with DCIS, the TBCRC and the RAHBT cohort. We analyzed a total of 774 DCIS samples from 542 patients with 7.3 years median follow-up. For outcome analysis, we used a subset of the RAHBT cohort and the full TBCRC cohort. So we had 97 RAHBT patients and 216 TBCRC patients here. We had cases who suffered subsequent ipsilateral breast events, either DCIS or invasive breast cancer, between 12 and 247 months from treatment, or controls who had no subsequent events between 18 and 243 months from treatment. For the Atlas, we have RNA sequencing, copy number variation, and mutational data, as well as protein expression by multiplex ion beam imaging. We worked with archival FFPE samples, which allowed us to perform spatially oriented analysis, and we were able to include a lot of patients with clinical information and long follow-up. The main goal of the case control study was to identify a prognostic signature. So we used RNA sequencing data from the TBCRC cohort and performed gene expression analysis to identify differentially expressed genes in cases with recurrence within 5 years from treatment versus the rest. And the reason we chose this 5-year cutoff was to avoid including non-clonal events, which may be more common in later years. This analysis identified 812 genes, and pathway analysis showed that these were involved in cell cycle, metabolism, MYC and mTOR signaling, as well as the p53 pathway. We then used the 812 genes identified in TBCRC to build a random forest classifier for predicting recurrence, that being either DCIS or invasive recurrence. And this was trained in TBCRC and then tested in RAHBT for validation. And of course, we get a very nice split between the predicted low and high risk group in the TBCRC training cohort. But we also see in the RAHBT test cohort that the area under the ROC curve is at 0.72, indicating good sensitivity and specificity. And we also observe a nice split between the low and high risk group from the model in the RAHBT test cohort. So this indicates that our 812 gene classifier also performs well in the test cohort. We then looked at how good the classifier was at predicting invasive recurrence specifically over the entire course of follow-up time. And here we found that our classifier was a significant predictor of invasive recurrence in both cohorts, both in Kaplan-Meier analysis and also in Cox multivariate regression analysis, where we adjusted for treatment. We also performed a multivariate analysis looking at either DCIS or invasive recurrence, where we also included treatment, age, ER by immunohistochemistry, and DCIS grade. And here we found that the only variable significant in both cohorts was the 812 gene signature. So this indicates that this classifier could be the basis for a future clinical test to assess outcome in patients with DCIS and stratify patients for treatment. Now that we're leaving the outcome analysis, in generating an atlas and having two large DCIS cohorts, this gave us the opportunity to investigate cluster solutions or subtypes for DCIS instead of just relying on the subtypes that are made for invasive breast cancer like PAM50 or the integrative clusters. And we used non-negative matrix factorization to cluster and identify a 3-cluster solution in TBCRC that was also found in RAHBT. And not surprisingly, ER and HER2 were among the main drivers of this clustering. So we identified the ER high group, which was characterized by high ER expression, the ER low, which had low ER expression, but high HER2 expression. And then our final group also had high ER expression, but lower expression of Ki67, GLOT1, amongst other genes, which indicating less proliferation and less metabolic activity compared to both other groups. So we labeled this group quiescent. We also performed unsupervised clustering of the DNA, and here we combined the TBCRC and RAHBT data together. We identified 6 subtypes based on copy number variations, and these were characterized by amplification that have previously been observed in high-risk integrative cluster subtypes from invasive breast cancer. Cluster 1 and cluster 4 showed amplification of the ERBP2 locus on 17q12. For cluster 4, this amplicon was larger and also spanned 17q23.1, which has been previously observed in integrative cluster 1 or IC1. Cluster 2 was characterized by amplification of HP1123, known from IC6, and cluster 3, 11q13.3, known from IC2. And finally, cluster 5 had amplification of 20q13.2, known from IC1 and IC9. Cluster 6 was a copy number aberration quiet subgroup, so this was characterized by the absence, or at least diminished signals of any of these copy number aberrations. And this was the largest group, which is in agreement with the overall lower genomic instability we observed in DCIS compared to matched invasive breast cancer samples. We also performed RNA expression analysis of LCM, or laser capture microdissected stromal samples in RAHBT. And here we identified 4 distinct DCIS-associated stromal clusters. We used pathway analysis of differentially expressed genes in each cluster versus the rest to describe the major characteristics, as well as maybe results to demonstrate protein expression and cell type variation in these clusters. So we had the normal cluster, which was characterized by extracellular matrix organization and focal adhesion, the collagen-rich, which was characterized by collagen metabolism and TGF-beta signaling, and also showed high expression of fibroblast-activated protein. The desmoplastic cluster showed fatty acid metabolism, high presence of CD8 T cells and myofibroblasts, and also high expression of smooth muscle actin. And finally, the immune dense, which was characterized by immunoregulatory pathways and showed high expression of CD45. To summarize, we have identified and trained an 812 gene classifier in TBCRC, which was validated in the RAHBT cohort. And this was found significant in multivariate analysis, including treatment, age, grade, and ER status. We also identified a new transcriptomic classification solution with 3 major subgroups in DCIS, as well as DNA-based cluster solution characterized by amplification seen in high-risk invasive breast cancer subtypes. And we also identified 4 stroma-specific signatures. For this atlas, we chose to work within the FFPE space, which of course is relatively inferior material compared to fresh or fresh frozen samples, but it allows us to work in a scale of hundreds rather than just tens of patient samples. It enables us to make observations that connect molecular findings with patient states and outcomes. And for this atlas, we brought together pathologic, molecular, clinical, and epidemiological ideas and expertise. And of course, the methods and results from this atlas can be applied to other clinical or private samples. Finally, and importantly, the data will be made publicly available through the HTAM portal. The results are presented here and more are available in these 2 papers. They are currently in preparation, but they are available in bioRxiv. And finally, I'd just like to thank everybody who worked on generating the Breast PreCancer Atlas. Thank you to our funding supporters, and also thank you to the San Antonio Breast Cancer Symposium for letting me present our work.